So it's time to talk about respiration. You really should take notes from this just as you would take notes in class. So right now you should get your notebooks out if you don't already have them out, or you should, if you have your notebook out, go to a new section and title it Respiration. Okay. This is the last topic in the chapter, and I'm not going to go over an immense number of details on this unit because I really don't want to burn you out. This can be a very difficult thing when you go over all the details. So it might seem like we're doing details, but really what we're doing is we're popping the hood and we're just identifying things and we're putting the hood back down. Respiration is not what you're probably thinking. Respiration to a lot of people is just breathing. That's not the respiration we're talking about right now. We're talking about cellular respiration in this chapter, and cellular respiration is for transferring the bond energy of fuel molecules like food molecules into bond energy of phosphates. So the food molecules can be stuff like this stuff can be glucose, or it could be triglycerides, or it could be diglycerides, or it could be sucrose. It could be a lot of different things. But the purpose of respiration is to get that energy out of those and put it into phosphates. It turns out, remember, that your organelles will only run on phosphates. So endoplasmic reticulum, the Golgi apparatus, the nucleus, the ribosomes, they only use phosphates for their energy source. Now, we're a eukaryote. That's because we have complex cells. But prokaryotes, this distinguishes from prokaryotes. Prokaryotes and eukaryotes have glycolysis, but most prokaryotes don't have Krebs and ETS. We do. All plants have all three. All animals have all three. But microbes only usually have glycolysis. So in eukaryotes, it says here, and that's us, that includes us, respiration consists of three phases. Turns out that in Biozone, it says respiration consists of four phases. I don't stress one of the four. It's called the link phase, or the link reaction. I'm going to let that out. I'll mention where it is, and I'll show you where it is in a little while when we get to the diagrams, but I'm not going to really stress that, and it won't be on the test, other than I might ask you, like which of the phases is the conversion of pyruvate to acetate, but that's getting ahead of the game. So let's talk about these. Prokaryotes and eukaryotes use glycolysis. Glycolysis is basically the beginning of respiration. It's basically cracking sugar in half. It takes place in the cytoplasm. Glycolysis is your process. You own it. It is a process that's ancient. All organisms on Earth can do glycolysis. Everything on Earth can do glycolysis. It takes place in the cytoplasm of all cells. Now, for organisms that have mitochondria, which generally are just the eukaryotes, that is, fungi, plants, and animals, and a few protists, these two processes take place in the mitochondria. The Krebs cycle is a bunch of reactions that take place on the inside of the mitochondria called the matrix. And the electron transport system takes place on the inner membrane of the mitochondria. So the one reaction that I don't talk about in respiration is the link reaction that links glycolysis to the Krebs cycle. Not a big deal, but I'll mention the reaction when we get there. Let's go ahead and skip to the next slide. Hang on a second. Let's see, cursor. Okay, go to the next slide, and there we are. Now, you might want to write down in your notes, or actually you really want to write down in your notes, this equation. The equation for aerobic respiration is right here. And don't worry about the delta H. This stands for delta, the triangles delta and H. That means that's the heat change. So what you want to write in your notes is C6 H12O6 plus 6O2 yields, that's the arrow, 6CO2 plus 6H2O. And the heat exchange for the reaction is 3,811 calories per mole of glucose. Now, I'm not going to ask you about the 3811, but you can bet that you probably have to write this equation on the test. The C6H12O6 plus 6O2 yields 6CO2 plus 6H2O. Basically, what this equation summarizes is the movement of energy from glucose to the energy of ATP. That's what this energy is going towards. ATP is the energy source for all of your organelles except your mitochondria. Your mitochondria make their own ATP by respiration, and then they give extra to you. So remember what Lynn Margulis said. Lynn Margulis was the scientist that said that mitochondria are separate life forms, or they were separate life forms, and they used to live independent lives, and now they live inside of us, and we sort of grow them. So back here on this other slide, we were talking about the Krebs cycle and the electron transport system. These two systems are only in eukaryotes, which are the organisms that grow their own mitochondria to produce most of their ATP. So that brings us to the next slide, which we've already seen. This is the slide that talks about the ATP-ADP cycle. So this should already be in your notes. We started the chapter with this. Glucose, now this can also be a fat, like a triglyceride. By the process of respiration, is used to produce ATP right here, which then is used by organelles for fuel. When the ATP is used up, the organelles return ADP and phosphate back to the mitochondria for recharge. So ATPs, ADPs, and Ps are constantly being recycled among themselves to make your organelles happy. The organelles, that's what this is talking about over here, are your ribosomes, your nuclei, your Golgi apparatus, your endoplasmic reticulum, your cell membranes with active transport pumps. They're all using the ATP, but they can't use sugar. Sugar is not an appropriate fuel source for them. ATP is. And so that's what this part of the chapter is going to talk about, is how do we make our ATPs. So that brings us now, let's go here. Let's go to the next slide. Glycolysis. Now you really don't want to write all of this information down, but there's a few things that are important here. So put a title in your notes, glycolysis. Glycolysis is basically 9 separate chemical reactions. You don't need to know the 9 reactions. But what they do is they split glucose in half, and they crack it into 2 molecules of pyruvate. So the glucose molecule, remember, is a hexagon. And what glycolysis does is splits that into 2 separate molecules of carbon, hydrogen, and oxygen, and each one of these is a pyruvate. So the glucose is literally split in half to do that. It turns out glycolysis uses a few ATP. So 2 ATP are required to start glycolysis, okay, 2. But it actually makes 4 more ATP. This is like real life. When you want to start a fire in the fireplace, you got to light a match, which is a fire, to get more fire. So the cell actually has to blow 2 ATP to get 4 back out. And the purpose of glycolysis really is not just to make pyruvate. It's actually to feed your mitochondria. But there are a few hydrogen atoms, which I won't stress, that are released by glycolysis, and they end up going to the mitochondria so that the mitochondria can use them to make even more ATP. So in summary, glycolysis is 9 reactions, splits glucose into 2 molecules of pyruvate. It uses 2 ATP, but it makes 4 ATP, and it throws out a few hydrogen atoms. And that's it. There's really nothing else to talk about when it comes to glycolysis. The next slide is actually going to show you the like a graphic that describes the 9 chemical reactions, and this is in your chapter text, but we are so not covering these chemicals here. But like I said, I summarized. See, there's an ATP getting used. Here's an ATP getting used. And then here's some ATP getting made. ATP getting made there, and there's some ATP getting made here. and there's a few hydrogens coming out. Actually, way back here, right here, there's a few hydrogens coming out. So, and then the final product is pyruvate. That's down here. But you do not need to know any of the details of a slide like this. That's way beyond the scope of what we want to do. This is an animation of glycolysis. This is a little easier to wrap your brain around. And we'll wait for the animation to start again. Here it comes here in a second. So here's glucose, hexagon. 2 ATPs get used. Then it cracks in half. Then some hydrogens are stripped off, and these NADs pick them up. And then all these P's that are on there, they're all picked up to make ATPs. So in essence, this is trying to show you the high points of glycolysis, which were glucose, 2 ATPs used, split into pyruvic acid, 4 ATPs get made. And that's the summary. We don't need to do anything more with glycolysis because it's, like I said, it's beyond the scope of what we need to cover anything else. Now, we already did fermentation in class. We actually did a lab where we fermented chemicals. But fermentation is something that we got to talk about briefly here because fermentation, as it turns out, will happen in any organism that can't get air. So we ferment, but we only ferment for a little while. We cannot live on fermentation alone. Fermentation is the production of ATP in the absence of oxygen. Basically, fermentation occurs anytime cells are deprived of oxygen, and the fermentation process is anaerobic. We call it's anaerobic, meaning it's without air. And fermentation is basically an anaerobic respiration. And what fermentation does is it dumps hydrogen atoms from NADs onto pyruvate. Now that sounds confusing, and I'll show you a graphic in a minute, but let's go back to the previous slide. Let's erase, and let's hit that previous slide. Here. Actually, 2 slides back. Here. Okay. So right here, in this step, see, here's the glucose. This is the hexagon here. That's a hexagon. That's glucose. This is pyruvate. That's half of glucose. Okay. Right here in the middle, there's some hydrogens released, and they're picked up by NADs. The catch is, normally these NADs go to the mitochondria, to mito. But if the mitochondria have shut down because they're not getting oxygen, these NADs cannot drop off their hydrogens there. In order to keep glycolysis running, these NADs here have to drop their hydrogens off to something. So what they do instead is they dump them onto pyruvate. That's what fermentation in the human body is. Pyruvate then in the human body gets converted to lactate or lactic acid, and I'll bet you've heard of that. Most people think it's bad, but it's actually good to be able to make lactate. Oop, there's a T there. It's good to be able to make lactate because lactate allows you to keep running when your mitochondria have gone anaerobic. When mitochondria go anaerobic, they don't work anymore. So they're not helping you at all. They're not making any of your ATP. So you actually crank out ATP using glycolysis and then dumping your hydrogens onto pyruvate, making lactate. It is a short stopgap measure in the human body. You cannot live on this process long. And eventually the lactate will build up and it'll hurt you. It makes you feel nauseated. You may throw up. People who run the 400-meter dash and track have a problem with lactate. Anybody who does heavy exertion exercise has a problem with lactate. But do recall, if it wasn't for lactate, sprinters wouldn't be able to run the 400. You'd run about 100 steps and then you would fall on the track because your cells would shut down. They would be unable to make ATP anymore to keep moving forward. What fermentation does is it buys you time. It keeps your muscles going, but in the end, you pant after you stop running because you have to pay back what is called the oxygen debt to your body to break down the lactate that you built up. So if I go back a couple of slides, I can show you what I'm talking about here. If I go back, let's see, 1, 2, go back to this slide. So if you're sprinting, Your pyruvate cannot go into the mitochondria. That's blocked because the mitochondria are shut down. You're not breathing fast enough to feed your sprinting muscles. So what you do is the NADHs that are picking up hydrogens, they dump them onto pyruvate. In so doing, that keeps glycolysis moving, and you make a colossal amount of ATP. But the problem is you're building up lactate in your cells. Oops, there. You're building up lactate in your cells. And when you stop running, this has to be broken down. And it's better for you to stay up on your feet and walk around so you can circulate the lactate back to your liver. Your liver will convert the lactate either back to glucose or it'll burn it using oxygen aerobically in its mitochondria. But either way, it'll get rid of the lactate. And then your nausea or your nauseated feeling will go away. So the purpose of making lactate is to keep you moving in times of oxygen crisis, when you don't have oxygen coming in, which is any time you sprint or overexert yourself. It keeps you moving. If you were walking down the street late at night, which you shouldn't be, but let's say you are, and somebody stops to say hi, I'm thinking you're going to run home. That would be the smartest move. And you're probably going to be running on anaerobics because you're going to run faster than your lungs can supply oxygen. But you'll make it home because of fermentation. The only catch is you'll be panting for 15 minutes or more after you're done running to pay back the oxygen you need to get rid of the lactate. So the purpose of fermentation is to make those few ATP by cellular respiration. But now we need to talk about a couple of details of this lactate or fermentation in general. There are a couple of different types of fermentation on planet Earth. We do lactic acid fermentation. So when we ferment, we make lactic acid. Lactic acid is a particularly foul-smelling chemical. If your armpits are sweaty and you smell them, you know what lactic acid smells like. Lactic acid is, we don't smell like lactic acid when we sprint because it's trapped inside of our cells, and we don't smell anything that's trapped inside of our body. But I've already talked about this. Basically, lactic acid fermentation is you're dumping hydrogens onto your pyruvate to make lactate. These guys can then go back and keep glycolysis rolling, and that keeps you alive. Lactic acid, though, is a poison. You need to get rid of it. It will lead to muscle soreness if you don't circulate it back to your liver quickly and get it broken down. But there's a more famous fermentation, and that is alcoholic fermentation. And you do need to be able to recognize, I won't make you write this, but this organism, Saccharomyces cerevisiae, is famous because it is the source of all human drinking alcohol on planet Earth. There are very few organisms that make ethanol alcohol on Earth, and Saccharomyces is one of a very few. Saccharomyces is a yeast, and if you don't give it oxygen, like you saw in class, it will live, and it will make carbon dioxide, and you recorded the carbon dioxide being made in your pipettes as foam. It also made alcohol in the pipettes, but we didn't do it long enough to create a whole lot of foam, or a whole lot of alcohol, I mean. But worldwide, alcoholic fermentation is a multi-billion-dollar industry. It's used in the production. Alcoholic fermentation is used to make everything from tequila to wines, beer, all hard liquors, including whiskey, bourbon, and other beverages. So I can't even think of all the things that are made, but it's just a colossal number. So alcoholic fermentation is an absolutely famous process using Saccharomyces yeast to make stuff. The difference between all the different kinds of drinks is not the yeast, obviously, it's what you feed the yeast. So in whiskey, you're using different things than bourbon, which is using different things than, say, with beer. I know with beer, they use hops a lot. Blue Moon also adds orange peels after fermentation to add flavor. Whereas some drinks, like a Japanese beverage, might use rice And some people use potatoes to start out the fermentation. Corn is often used to make whiskeys, etc., etc. Depending on what you feed the yeast, that will determine the flavor of the alcohol and beverage that the yeast produce. So fermentation is a huge industry. I'll bet some of your parents actually work in the beverage industry, and it's basically based on yeast. So now we move forward. The importance of fermentation, as I said before, it's a temporary survival strategy. If you couldn't ferment, the moment your muscles were deprived of oxygen, you would faceplant on a track or on a sidewalk trying to run. But here's the catch. You can only ferment for a minute or two before you're literally out of breath and you have to stop running. We will die if we don't get oxygen after several minutes. So if you fall in a lake and you're not at the surface breathing within several minutes, your body will be fermenting, but it can't go on that for long. But that's better than only seconds. If we couldn't ferment, you'd be dead in seconds being underwater. But we can go minutes. In fact, donated organs can last many hours on fermentation alone, up to 20 hours for a kidney, say, to fly it from one city to another. So fermentation is a big deal. I should also mention the baking industry uses carbon dioxide from yeast to make bread rise. So bread is basically like a solid wad of starch and oils, but you basically mix yeast into the bread, and you knead the bread, which means you push the yeast into the bread and you roll the bread and you smack it around, and then you put it in the oven. And the yeast make carbon dioxide and alcohol in the bread, but the alcohol is such a small quantity because it only goes on for a few minutes, like 20, 30 minutes, that the alcohol also just evaporates away in the oven. But the carbon dioxide made bubbles inside the bread, and that's why breads are fluffy. That's where the bubbles come from. The yeast makes the bread rise. So there is no other way to do it. We would need to somehow come up with some other way of making little bubbles inside of bread, and that's just not an easy thing to do. Now, the last parts of respiration, I'm going to use diagrams and animation for, but I'll just emphasize what some of this stuff is now. The Krebs cycle and ETS can only take place if a cell is getting oxygen, and they take place in the mitochondrion. And you need to draw a mitochondrion in your notes, and I'll just show you a picture in a second. The mitochondrion contains two membranes. The outer one is smooth and like an oval, like a bean, and the inner one is rough, like a bag that's all wadded up inside. The fluid that's inside the inner membrane is called the matrix. And the Krebs cycle basically happens in the matrix, and the electron transport system and chemiosmosis all happen on the inner membrane. So on the next slide, you're going to see a mitochondria, and I urge you to go ahead and draw one in your notes so that you have like a drawing of how this thing is structured. So I would draw a mini mitochondrion that looks kind of like this. I'll draw it up here. So draw an outer smooth membrane, an inner rough garbled membrane, like that. And then this is the M for matrix, and that right there is the IM for the inner membrane. This is the OM out here, outer membrane. So I would draw a simple mitochondria in your notes. It doesn't need to have granules, ribosomes, cristae, and all that stuff. Not even the intermembrane space, not stressing it, not DNA or anything else, but the inner membrane and the matrix, M-A-T-R-I-X, that's kind of important. So with that in mind, we go to the next slide, which is begins an explanation of what the Krebs cycle and what these processes are. So the Krebs cycle was first documented by Sir Hans Krebs. He was actually an English or British knight. The Krebs cycle is a process that breaks down pyruvate to release energy. The Krebs cycle basically takes pyruvate from glycolysis and breaks it down to carbon dioxide and hydrogen. Now, I don't know that you should write all this down because I'm going to emphasize it on the diagram. But the diagram is actually demonstrating this. A little bit of energy um, is thrown into ATP, but the ETS and chemiosmosis make most of the ATP. The Krebs basically feeds the ETS material it needs to make the chemiosmosis happen and make ATP. So on these next few slides, you're going to see, like, this is the Krebs cycle. We are so not knowing all the details of the Krebs cycle, but it is complicated, and it involves basically pyruvate. This is the link reaction, the conversion of pyruvate to acetate. This is acetate here. Let's erase that. I just kind of obliterated it. This is pyruvate. Let's use the pen instead. There we go. This is pyruvate. This is acetate. It's the conversion of a carbohydrate basically to a tiny fatty acid. That's called the link reaction. We kind of leave that out in introductory bio. But the acetate then enters the Krebs cycle. And what happens is this is a 2-carbon acid. This is a 4-carbon fatty acid. When you add a 2-carbon fatty acid onto a 4-carbon fatty acid, you get a 6-carbon fatty acid. The 6-carbon fatty acid is literally rearranged, and then finally a carbon is ripped off, and it becomes a 5-carbon fatty acid. Then another carbon is ripped off, and it becomes a 4-carbon fatty acid. Then the 4-carbon fatty acid is recycled back into the starting 4-carbon fatty acid, and that's why it's a cycle. It makes something that it uses. So we're not going to know, though, this detailed diagram because it's beyond the scope of what we have to know to do that. So we're going to go instead to another diagram. But first, this is a basic animation of the Krebs cycle. It starts on the left here. So you see this? This is a 6-carbon acid. Some hydrogens get thrown off. Then it becomes a 5-carbon acid. And some more hydrogens get thrown off. Then it becomes a 4-carbon acid. And some hydrogens get thrown off. And then it's recycled. It's basically being recycled back to its original acid. Here's the acetic acid coming in. Acetic acid, 2-carbon acid, plus a 4-carbon acid makes a 6-carbon acid, which then becomes a 5-carbon acid, and then a 4-carbon acid, and it's recycled. So you might wonder, what in the world is the cell doing this for? Well, ultimately, the purpose of the carbon dioxide is to spray out hydrogen. So if you had to write anything down in your notes, the Krebs cycle's big job is to spray out hydrogen atoms so that the electron transport system can use them. All this other stuff like the CO2 waste, yeah, that's waste. I mean, so the Krebs cycle does make the carbon dioxide that you're exhaling right now in your breath, but that's not the purpose of the process. The purpose of the process was to crank out hydrogen atoms to feed to the electron transport system. Which brings us to the next slide, the ETS. The ETS happens on the inner membrane of the mitochondria. If by now your head's not hurting, you really need to go into biology because this is definitely your thing. The electron transport system is basically 4 clusters of proteins. They're called complexes. The ETS proteins are on the inner membrane of the mitochondria. They basically take the hydrogen atoms from Krebs, and they pass the electrons along. The individual proteins that pass the electrons are called cytochromes. And by the way, there are 4 basic complexes in the ETS, and the complexes are made of cytochromes. The purpose of the cytochromes, I'll just draw them in a sequence, just 4 circles. Hydrogen from Krebs goes on to these, and they pass along the electrons. As they do, they pump, let's go this way. They pump protons into the space between the inner and outer membrane of the mitochondria. Now, this can all happen as long as oxygen takes the electrons off at the end. So the way the electron transport system works, it's a series of 4 proteins that take hydrogens from the Krebs cycle. As the electrons from the hydrogens are passed along from protein cytochrome to protein cytochrome, they pump protons, protons, positively charged nuclear particles. They pump them into the space between the inner and the outer membrane of the mitochondria. Those protons are then used by a special whirlybird enzyme to make ATP, and that was the whole purpose of all of this craziness, was to make the ATP. At the end of the chain, oxygen is needed to actually take the electrons off. If oxygen is not available, the ETS stops, pumping stops, and the mitochondria shuts down. At that point, you're fermenting, and your body is praying that oxygen will be restored because you can't keep doing that for long. You must respire aerobically to survive long term as a human being. That brings us to, that's what this graphic is trying to show you. Here's the Krebs cycle. They call it the citric acid cycle here, but it's basically the same name. Krebs, or it's the same process, different name. This is the Krebs cycle. It squirts out hydrogen here, and that hydrogen is used to pump protons. See, these are the protons that it's pumping. There, there, there. Those protons are then used by another enzyme. This is ATP synthase. It's used by that enzyme to make ATP, and that's where most of our cellular energy is coming from. So let's wipe the screen here, and we'll go to the next slide. Let's see, I'm going to go to cursor. All right, now the last of the processes here is chemiosmosis. Chemiosmosis, and the Biozone book doesn't really count this as a process, but it really is. Technically speaking, there are 5 basic processes of ATP production in cells. The first is glycolysis, the second is the link reaction, the third is the Krebs cycle, the fourth is the ETS, and the final and last one is chemiosmosis. Chemiosmosis is where the protons are used by ATP synthase to form ATP. The most ATP you get out of a single glucose is about 38, and about 32 of them come from chemiosmosis. So out of every 38 ATP that you make, 32 of them are from the process of chemiosmosis, which is happening on the inner membrane of the mitochondria. The enzyme is very similar. If you remember from photosynthesis, there's a whirlybird enzyme on the thylakoid membranes that makes ATPs using protons. Well, you make your ATPs the same way. You use protons, which are hydrogen ions. That's all a hydrogen ion is. A hydrogen ion is a hydrogen atom without its electron, which is just a proton. You basically, with your whirlybird enzymes, you use the protons to crank out ATPs, a bunch of them. You can't live without this part of respiration. It cranks out 32 of the 36 ATP that you actually need from sugar. Now, the mitochondria don't give you all 32 of those ATP, but they give you most of them. They keep some of them for themselves. Remember, Lynn Margulis said mitochondria are their own thing, and we're just growing them and feeding them so that they'll help us. But they give you most of them. This is the whirlybird enzyme. There's spinning. This is the spinning part here. And there are 3 active sites. And this little stator rod actually does the bonding of the ADP and P. And this thing spins. So as ADP and P are loaded in the active site, this spins around. And this creates the bond between the ADP and the P to make ATP. So it's a really complicated enzyme, and biologists have spent a long time trying to figure out how this thing works, because it's at the heart of our existence and all multicellular life's existence. Now, I would urge you to draw this diagram in your notes. And what you want to start with is use up half of a sheet of paper. You're going to make a basic arc across the page. That arc right here, that's the outer membrane. This is the outer membrane of the mitochondrion. I'm going to have to go to pen and not highlighter. It's the outer membrane of the mitochondrion. And the purpose of the outer membrane, this is the OM, outer membrane. This is the IM, the inner membrane. The purpose of these 2 membranes is to create this space, which is labeled by number 9. And that's where the protons accumulate to be used by the whirlybird enzyme, which is right here. So on the outside, above, you want to draw a big arrow going from left to right, like that. Okay? On the inside of this oval, you want to make a wavy membrane like this. That's your inner membrane. Again, here on the outside, we've already drawn our smooth outer membrane. Then on the very inside here, this is the Krebs cycle. Okay, so we have Kc here in the inside. We have the ETS right here at number 3, ETS. And we have glycolysis out here. I'm just going to call it GLY on the outside. So again, one more time, you have, in fact, let's go to a white screen. Let me erase the screen. I'll draw it for you just on a white screen, and then I'll have to erase it and go back to the other one. There's the outer membrane. There's the inner membrane. You want to draw cycling arrows in here. That's the Krebs cycle. Out here, we got this big line going across. That's glycolysis. This is glycolysis. The electron transport system's on here, ETS, and this is the KC, the Krebs cycle, right there. Okay, and then you want to label these. This is the inner membrane, the IM. This is the OM, outer membrane. And we're talking about, like, pretend that the whole screen is a cell. This is cytoplasm out here. Okay, so out here is cytoplasm, but the mitochondria down here is an organelle, and it contains these parts. So let's erase the screen. Let's get rid of the white screen and go back to this. So let's go over what these are. Number 4 is glucose. This is a hexagon here. You know, glucose molecules are hexagons. Remember, they got the little flagpole, and there's an O, like right there. So that's glucose at number 4. And basically, you see glucose is broken down. I wish I could ask you this and give you a bonus. Number 5 is, what do you think that is? That is pyruvate. This is half of a glucose. So number 5 is pyruvate. Okay. And 10, you might have guessed it, 10 going in and 10 coming out. That's ATP. Remember, 2 ATP get used by glycolysis, 4 more get made. Now, before we dive into the mitochondria stuff, remember, if you're not breathing in oxygen, the pyruvate's converted to lactate, and that's where fermentation takes place. Yeast convert it instead to alcohol if they don't get oxygen. By the way, you never want to aerate your yeast solution. Never bubble it like a fish aquarium, because if you do, you ain't getting any alcohol. The yeast will just aerobically respire, and they won't make any alcohol, and instead they'll just make carbon dioxide and water, and that's not going to make your beverage make any money for you. Now, the pyruvate normally that you make, you feed it to your mitochondria, so in it goes. So number 5 is pyruvate. This is number 5 here also, that's pyruvate. In the mitochondria, I skipped the link reaction, but it's right here. Basically, the link reaction is carbon dioxide is released, and actually this would be like a different number. This would be acetic acid. Okay, let's erase all this and I'll start over. This is acetic acid, ACETIACID. That's the link reaction happens right inside the matrix where pyruvate's converted to acetic acid. So acetic acid basically reacts with a 4-carbon acid, and it becomes a 6-carbon acid, which becomes a 5-carbon acid, which becomes a 4-carbon acid. But as it does that, it cranks out hydrogen that's picked up by the NADs and the FAD. That's the main reason for the Krebs cycle, is to kick out the hydrogen atoms. So the glycolysis basically made pyruvate that feeds the mitochondrial Krebs cycle what it needs. The Krebs cycle cranks out hydrogen to feed the ETS what it needs. These hydrogens go to the ETS. They dump, or the hydrogens are dumped onto the ETS. The ETS pumps protons. Number 9 is protons. You know, H plus is protons. The protons are used by chemiosmosis, the whirlybird enzymes at 12, to make ATP. So ATP is number 10. Now, at the end of the ETS, you got to have oxygen, which is 6. 6 is oxygen, and you make water, H2O. So I'm going to erase this and go over this one more time. Let's start at the beginning. On the left-hand side, this is glucose. And this is ATP coming in and ATP going out. And this first, number 1, is referring to glycolysis here. Number 5 is pyruvate. Pyruvate is normally with oxygen present fed into your mitochondria. It's converted to acetic acid, which is a 2-carbon fatty acid. The enzymes then react the 2-carbon fatty acid with the 4-carbon fatty acid to make a 6-carbon fatty acid. The 6-carbon fatty acid has hydrogens ripped off, carbon ripped off. It becomes a 5-carbon fatty acid. Then the 5-carbon fatty acid has carbon ripped off and more hydrogens ripped off. It's converted back to a 4-carbon fatty acid, which is what we started with. And the hydrogens then go to the ETS. They're dumped off so that they can be used to pump protons. The protons get pumped, so 8 is hydrogen atoms, 11 is carbon dioxide waste, 2 is the Krebs cycle, 10 is ATP. The Krebs cycle does make a few ATP. In the ETS, 8 is the hydrogen atoms again, 9 is the pumped protons, 3 stands for ETS here, 7 is basically water being made at the end of the electron transport chain, but to do that, the cell uses oxygen, or the mitochondria uses oxygen to do that. And then, using all the protons at number 12, we have chemiosmosis is used to make ATP. That is aerobic respiration. That's what you'll need to know for the test. This diagram is on your test. It's also on your biosign, so you should study this diagram. It is the good summary of aerobic respiration. In just a second, the animated version of this is actually going to be on the screen. So I know this looks confusing, but we really have only scratched the surface here. As far as, by the way, as far as ATP yield is concerned, glycolysis makes a couple of ATP, Krebs cycle makes a couple of ATP, but all the rest of your ATP are coming from chemiosmosis and the electron transport system. Actually, just from chemiosmosis. So the way respiration works is glycolysis gives the Krebs cycle what it needs. The Krebs cycle gives ETS what it needs. ETS gives chemiosmosis what it needs. Chemiosmosis gives the organelles what they need. So, and which is ATP energy. So now the total animation, I believe, is on the next slide. Let's see. Yes. Now this actually is the end. This is just a graphic. This is glycolysis on the left. Here's pyruvate right here. Pyruvate is being fed into the mitochondria. Here's the Krebs cycle. Here's the hydrogen that's being sprayed out. The hydrogen then is being picked up by the ETS, and the ETS is using this whirlybird enzyme. I mean, it's pumping protons so that the whirlybird enzyme can use the protons and make ATP. More important than this, though, is the graphic, and I don't stress this is the last slide really of the chapter, because the last slide here is just a summary, and I'll go over that in a second. Here's the last slide. Let's see if I can't restart this, if it'll do that for me. Click this. Nope, I'll have to wait for it to come around. So let's watch this as it comes around. It'll start on here on the upper left. So this is your mitochondrion and your glycolysis diagram you drew. Here it comes. Here's glucose. A couple of ATP are used. This is glycolysis on the top here. Okay, glycolysis cranks out pyruvate. You're going to see pyruvate here. There's pyruvate. Here's the 4 ATP made by the glycolysis. Then the pyruvate is sent in the mitochondria. It's converted in the link reaction to acetic acid. The acetic acid reacts in the Krebs cycle to form a 6-carbon acid, then a 5-carbon acid, then a 4-carbon acid. And all the while, hydrogens are being sprayed out and picked up by the ETS. And you'll notice the ETS is pumping protons, which makes most of your ATP. It was very fast, but right here at the end, you had like 34 ATP sitting there being made. So it's kind of a big deal. Glycolysis directly nets 2 ATP. The Krebs cycle nets 2 ATP, and I believe actually it's not 32. In a really optimized cell, you'll get 34 ATP from all the hydrogens that are sprayed out by the Krebs cycle and a little bit by glycolysis. And so right here, you're going to see the ATP being made right here. Here it comes. There they are. My green pen had it covered up. So that's it. That's the overall process of cellular respiration, starting with glucose and glycolysis, making pyruvate in glycolysis here. The pyruvate link reaction go to vinegar, or acetic acid is another name for vinegar. The vinegar enters the Krebs cycle, becomes a 6-carbon acid, then a 5-carbon acid, then a 4-carbon acid, and all the while sprays out hydrogen. The hydrogen is used here by the ETS to pump protons. You can see we're up to 48 protons, now 56 protons, then 68 protons. Those are used to crank out your ATPs. And that is kind of the end of the process of cellular respiration. Now, the last thing to talk about is the summary here. And I wish it was easier than this, but we did this for photosynthesis also. In respiration, your glucose energy is transferred into hydrogen atom and ATP energy. The hydrogen atoms are used to move electrons. The electron movement creates a proton pool. The proton pool is used to make ATP energy, and the ATP energy is used to do your organelle work, like ribosomes use it to make proteins, the nucleus uses it to make DNA, the cell membrane uses it to do active transport, et cetera, et cetera. So to summarize one more time, glucose energy becomes hydrogen atom energy, becomes basically a proton pool, which becomes ATP energy. That's what the purpose of glycolysis, the Krebs cycle, and the electron transport system. Glycolysis takes some of the glucose energy and puts it into some hydrogen atoms, but more importantly, the Krebs cycle puts glucose energy, it's really pyruvate, but pyruvates are half glucose. It puts the energy into hydrogen atoms. The ETS takes the hydrogen atoms and moves electrons and then pumps protons. And the pumped protons are used to make ATP. And that is used, the ATPs are used by your organelles to do things. So that concludes your notes on cellular respiration.